to physics of behavior. Very happy to see so many of you. There's places also up front here. So I'm going to start right away with our first feedback loop. Okay? And it goes like this. We human beings can be in different states, and one of them, let's call them the relaxed state. Welcome, everyone. And in the relaxed state, we're good for listening, for learning. It's a state we want to be in this course, hopefully. Yeah. And when we're in the relaxed state, our body does certain behaviors. For example, our breath, we have deep kind of breaths, deep breathing. Now, since we're human beings, we have an amazing thing we can do. We can take a deep breath, or breathe deeply, and that increases the probability that we get into the relaxed state, which has better listening, concentration, memory. And so we can all do this. It's like, we all know how to do it. It's like taking a big, deep sigh of relief, like this. We all know how to do this, right? When we're relieved of something, like that. So I propose in this course, from time to time, we'll practice this. And you don't have to do it, but if you do, I guarantee you'll enjoy it. Take a nice, deep sigh of relief like that. Yeah. So if you feel like it, let's all together take a nice, deep sigh of relief. And when we have people coming in late, of course it's the first time, we're going to honor them, or just make sure we recognize them by taking a nice deep sigh of relief in their honor, like this. It's not to embarrass you, just so that you can find your place. So my name is Uri Alon. I'm a professor in biology. And this course, called Physics of Behavior, we will explore a way of thinking about biological systems. The goal is, by the way, I wrote down here what we're going to do, right? So welcome, breath, we already did 2 things, great. The goal here is that by the end of this course, you will think differently about biology and learn to see inside the complexity that goes on inside our cells, a certain kind of simplicity, which is principles, which are mathematical principles that can help you connect different kinds of systems. Let's take a nice deep sigh of relief. Welcome. Connect different systems on universal principles. It can also help us make sense of what those systems are doing, what their function is, how they evolved. So that's the goal in this course. And we'll use both biology and mathematics, and I recognize that this class is very heterogeneous. So just to find out who is here, I want to ask you to turn to someone next to you that you haven't spoken with yet and find out who they are, what their name is, and what they study. What's their field? Especially, let's say, what they studied in their undergraduate degree. Okay, find out from each other. So let's do that for a second. It could be behind you too. Yeah. It could be behind you. גם אתה יכול גם למצוא מישהו, למצוא בשלישייה. Okay, thank you. Now, my question to you is, it's fun, it's hard sometimes to stop people when they're talking, it's so fun. My question to you is, how many of you found somebody who studied something different from you? Okay, so that's a huge amount. Let's do the control. How many found somebody who studies the same thing as you? Okay, thanks for being brave. So we have a lot of different people here, and instead of that being an obstacle, I want to propose that this could be a resource. My goal is also that we help each other learn. I'll soon say what, But I just really want to find out what disciplines are here. So what are you guys studying? So who's from physics? So about half of the people. Who's from biology? Okay, so like a few people. Who else? Computer science, raise your hand so I can see that. It's important for me to know. Yeah, who else? Chemistry. Okay, like 5, 6 people. Who else? Neuroscience, can you raise your hand? Okay, welcome. Yeah. Who else? Math, can you raise your hand? Thanks, it's very useful to know. 3 people I saw. Who else? Any social science? All right. Great. So when I say heterogeneity, I mean that I'm going to say in this course sometime words from biology like this. What's the difference between transcription and translation? So biologists, this is like breathing air. Yeah, it's like obvious. For other people who haven't studied biology, maybe we need to learn this. So I'm just wondering, who for you, it's like absolutely obvious. What's the difference between transcription and translation in biology? Just raise your hand. Don't worry. Okay, good. So, and then we'll also have equations. Now I just want to say again, maybe so you understand this. My goal is that you understand what this course is, if it's right for you. We're not going to do really heavy math here. If you like big, complex mathematical formalisms, this is not the course. We're going to do as simple a math as we can to understand what we need to in biology. So, for example, we'll use equations like this. So for some of you, solving this equation, like breathing air. For others, you need a little to refresh. Okay? So if you guys, if this solving this equation is like breathing air for you, raise your hand. It's like, okay, so we have, good. So so this just to orient, I'm trying to orient you what to expect here. Okay, so a little bit more information before we start talking about biology. Rules. So the website has some rules. I'm kind of I want to say first, I want to welcome people who are just auditing without doing the exam. So you're welcome here. Those of you that want to take the exam and want to get a grade, we'll have an exercise every week, and not this week, but next week it will start. This exercise will, again, the goal is not to give you a lot of work to do, but to train the way of thinking that we do in this course. We have a teaching associate, Avi Mayo, who's over there. And maybe based on the size of the class, maybe we'll get another one. And we'll have an open hour that you can come and talk with Avi and graduate students in my lab, which is Sunday from 10 to 11 in Wolfson, 612. And this Wolfson building is the big biology building, the biggest biology building. So you can come and ask questions over there. And we'll have an exam also in the end. Oh, we don't need this. Yeah. Yeah. Yeah, also, I'm going to use the whiteboard. It's also important to say. And the reason is, welcome, let's take a nice deep sigh of relief. Because, as you'll see, it creates a kind of rhythm and interaction that I like. But then you can ask, if there's no PowerPoint, how am I going to know review the material? So there's some resources. You see there's a nice photographer here. Yeah? Can you say your name?
Hi,
Itzik. And Itzik was also photographing in 2014. So there's the video of a course from 2014 that has a lot of what else, 2000, not yet 2104. Yeah, 2014. בסדר. בסדר. On my website. And another resource for you is this book, An Introduction to Systems Biology, Design Principles of Biological Circuits, that is available in the library and covers a lot of the material. All right. Any questions about this part of which we call, let's say, rules and resources? for us.
Soft copy of the book? Is there?
The question is there an electronic copy? No, there's no. Okay. We need to lift this up. איך? Zole, okay. So while it's going up, let's take a nice deep sigh of relief. We successfully completed this first part of orientation. Good for us. Okay. And now, let's start talking about physics of behavior. So in physics, a lot of times we like to look at the simplest system that has some properties, like the hydrogen atom, like the harmonic oscillator. And we're going to talk about a simplest system for us will be a cell. We'll talk just in this lecture about single-cell organism, bacteria called E. coli, lives here inside us, in order to demonstrate how cells think, you can say. So this cell looks like this. Size 1 micron. It's really a fully functioning living organism. You put in some water with some salt and sugar, and after 30 minutes, it copies itself exactly, including all the millions of letters inside its DNA, basically without error, making maybe one letter error every 300 times it divides. If you put some sugar over there, it senses it. It grows an electric motor that spins at 100 hertz with a propeller and a navigation system that tells it where to go. Despite Brownian motion that changes its direction by 90 degrees on average after 10 seconds. So it's tiny, but it knows where to go. It grows pumps to take that sugar in and break it down to carbon atoms so it can make itself. This one cell. And all these things I mentioned so far, this motor, those pumps, etc., are molecules that the cell can make, which are called proteins. Size, nanometer, so much smaller. So you can have a pump, an enzyme, look like a Pac-Man. You can have a little motor made of many kinds of proteins, flagella, a navigation system made of proteins. So proteins are molecules that do things in the cell. They're really amazing. They're made about 10 to the 4 atoms, but they're not random polymers. They're built like machines that can transition between states, active, inactive. So E. coli can make there's 4,500 kinds of proteins, and a total of about 1 million copies of proteins per cell. So some of those proteins are found in low concentrations, maybe 10 per cell, and others are found at high concentration, 100,000 copies of the same protein per cell. So E. coli changes its composition according to the environment it's living in, environmental signals. And that's something we'd like to understand in this first lecture, how it does that, how quickly it does that, and what's the brain that makes this computation. I say brain, of course, it doesn't have neurons like us. Each neuron in our brain is a cell, much bigger than E. coli. It has a chemical brain that does that. Okay, so we talk about cells and proteins. And I like to call this sometimes the cognitive problem of the cell. And to say it like this, E. coli lives in a world that's, sense, or it's sensitive to many, many kinds of things about the world it lives in, signals. Things like, pH, temperature, hundreds of different kind of nutrients, toxins, gradients. Let's take a nice deep sigh of relief. You're here. Welcome. And the question is, how can E. coli compute what is the best composition to make for each environment it's in? Because again, these are life or death decisions. If it doesn't make that pump when it has the sugar, it won't be able to import it, grow, divide. Maybe another cell that can do that is going to divide, grow exponentially, take over the test tube. So this is a race for life or death for E. coli, to make the right decisions given its environment. How does it do that? So again, this introduction here is heavy on biology, just to get everyone oriented, and then we'll get to some equations. So how does E. coli make a protein in response to the signal? So for example, if a sugar is present, how does E. coli know to make the pump? So the proteins are encoded in genes on the DNA. So I'm going to draw here a gene. This gene, in order to make the protein, there's a protein called RNA polymerase, which is the protein that can bind the DNA here next to the gene. So this is a binding site. Binding site is a little sequence of letters in the DNA that this protein RNA polymerase is built to recognize. And once it binds there, it copies this gene into a little scratch pad note that's quickly made and quickly destroyed, mRNA, that has the same information as the gene. So this is called transcription. And then this mRNA is made into the protein. So if this is gene for protein Y, then RNA polymerase binds, makes the RNA, makes the protein. This process here is called translation, carried out by a big factory called a ribosome. Now, this picture would mean that Y is made constantly. But how does Y, let's say Y is a pump, know how to be made only when the sugar is there? Yeah? And so the way that works is that there's I'm going to say here that cells encode, or let's say it like this, represent the environment inside the cell. So an internal representation of the external world by about 300 special proteins. These are information-carrying proteins called transcription factors. We'll call them transcription factors, TFs. So we have these environmental signals, like S1, S2, S3, this could be sugars, etc. Maybe also internal things like my DNA is damaged. Yeah? They cause the activation of a set of 300 transcription factors in E. coli. And these transcription factors go and regulate the different genes on the DNA. Gene 1, gene 2, gene 3, like that. So the cell, inside the cell, there's like 300 degrees of freedom, which represent the many, many more degrees of freedom in the external world. And evolution designed these 300 degrees of freedom to care about what's important for the cell. Like one of them cares about DNA damage, another one about sugars. And the way this works is the following. So you have a transcription factor X here. And like I said, like many proteins, this protein is a molecular machine that can be in two conformations, active and inactive. And it transits between these conformations quickly in a microsecond time scale. And when, in our case, a sugar is there, it binds to this protein and makes it active, makes it more likely that it goes to the active conformation. Then this protein finds on the DNA its specific binding site. So up near the gene is this whole region here, which is called the promoter. It's a regulatory region that guides when this protein is made and when it's not. And X finds this site, searches the DNA, you can say finds this site by diffusion, binds there. And when it binds there, it increases the probability that RNA polymerase will do this. So this little biochemical circuit starts with the signal, let's say sugar inside the cell, binds the transcription factor, causes it to change into its active conformation. It diffuses in the cell, finds the promoter, helps RNA polymerase bind there or be more active. Welcome. There's a place up here if you want to. Take a nice deep sigh of relief. Here. And increase the probability that Y will be made. I just want to say, and this entire thing here, this entire biochemical detail, we're going to symbolize in the following way: X, arrow, Y. And maybe we'll add this SX here, which is the signal for X. So this will be our symbol for this interaction. I just want to say that there's, okay, so we talked about the cognitive part of the cell, signals, genes, transcription factors, RNA polymerase, promoter, activator. I can say X can increase Y production. In that case, we'll call it an activator. And there's also kinds of transcription factors that decrease. That is to say, by binding, they interfere with RNA polymerase. And then we call these repressors. And sometimes we'll denote repressors by this kind of arrow. And this is this kind of arrow. This is a repressor, this is an activator. There's a very neat thing that evolution gave us, like a gift. As scientists coming to study how the cells think, and that is that the different reactions I just talked about have very strong separation of timescales. The timescale here in these transitions is microseconds. The timescale to find the DNA by diffusion, like that, is on the order of seconds. These reactions here takes minutes. And as we'll see later, the time for protein Y to accumulate in the cell to reach functional levels can take an hour, hours. So that means each step here occurs on a really different time scale. And when you have that property, it's very useful, separation of timescales, because when you write a mathematical model, you can assume that slow reactions are not changing while fast reactions are changing. And that gives us a lot of power to make mathematical models. So in this lecture, all of these stuff that happens on this time scale, microseconds, second, minutes, etc., we're going to chunk in to an instantaneous event and say, okay, protein Y has started to be produced or stopped to be produced, instead of needing to worry about all these timescales and still get a very accurate answer if the timescale we're interested in is hours. Did I explain myself? Yeah.
Can you please say again the difference between S, X, and X star?
So the difference between SX, X, and X star. Okay. So X is a protein, a transcription factor protein, and it's built to detect a signal like sugar, and other transcription factors can detect DNA damage and say, oh, my DNA is damaged. Let's find a gene that makes DNA repair proteins, for example. So there's different kinds of signals. Did I explain?
Yeah.
Yeah.
What did it start?
What is?
X star.
X star is the active conformation of the transcription factor X. And maybe I should say here that proteins, a lot of times, are molecular machines that can change between two conformations. This is a really shape, and the time scale is microseconds. And let's say this shape here can recognize the DNA binding site. And thermodynamically, it has lower free energy for this reaction. Okay, and X also has a little pocket for binding SX here. So all this stuff is fascinating. You can write theory for it. It's not going to be our main topic, but yeah.
Is there many copies of the DNA inside the cell?
Okay, thank you. So, you know what? I have an idea. Because these questions are excellent, and when you're asking them, and thank you for asking them, because I'm guaranteed there's other people here asking the same questions. So I'm going to, some people though maybe have a question you don't want to ask right now, but it could be nice to get an answer. So let's do the following. Like I said, we're heterogeneous. Some of the people here really know the biology I'm talking about. Can you raise your hand if it's obvious for you? So look around you. If you look around you guys. And what I want to ask you is to turn to someone next to you, like that, and ask them the question, like this question. Okay? Ask them that question, and we'll spend a couple of minutes just, what do you want to know more from the biologists here about what I'm talking about? So, and you can also, if you don't want to do that, you can just listen to a conversation like that near you. Okay, so let's spend like 3 minutes doing that. So please go ahead. יש לי איזה כיף בכתף. יש לכם איזה שאלה? לא, נכון. משהו? שאלה? נכון? איזה שאלה? אתם ביולוגים? פיזיקאים? ואת? כלכלה? יפה מאוד, טוב מאוד, טוב מאוד. זו הזדמנות לדבר עם פיזיקאית ביולוגית, זה נורא נדיר. יש פה משהו יכול להיות שיש פה משהו מה זה?
ברוב הקורס אתה הולך להראות איך ממדלים מנגנונים מולקולריים ביולוגיים או התנהגות גם ברמת אורגניזם או בן אדם?
זהו, אני שיניתי את זה לפני 3 חודשים, שיניתי את הקורס למנגנונים ביולוגיים. זה פחות מתאים לך?
לא יודע, רק לדעת.
כן. אוקיי. So thank you for that. So yeah, as far in E. coli, it's basically one DNA per cell. I'm just curious, did anybody here discover some interesting fact that maybe can help, like in this conversation? Something interesting? Yeah.
I understood that there is binding site in every gene, but not every gene has a regulator binding site that it is.
Okay. So let me repeat that. Every gene has RNA polymerase binding site, so it can be transcribed, but not every gene has a regulatory binding site. Each kind of transcription factor only regulates a few genes. Yeah. So each gene has 0, 1, 2, 3 binding sites for different transcription factors, so it can respond to 0, 1, 2, 3 signals. In E. coli, average is 2. In human beings, you can have genes with 20 regulators like that, carrying out a program like X1 and X2, but not Y1 or Y2 and Z bound for more than 30 minutes, and with a probability of 30%, if like this, a program like that in the promoter, which guides the decisions made from a little cell into our whole body. Like, how does this know to be a nerve inside there, a nerve, a muscle, like these kind of, so these programs, amazing programs. We stick now with E. coli, which is a simple world, but has a lot of the same principles as human being biology. Human being has some additional complexity, but isn't that amazing, by the way, that bacteria and human beings have such basically similar, conceptually, issues. Any remaining questions right now?
But does the same gene appear more than once?
Inside the DNA, does the same gene appear more than once? Usually not. There are some exceptions.
You said that in E. coli, every DNA appears once.
Yeah. So
But
it's
in other cells,
in other cell types, like in human beings.
or other bacteria.
Yeah, other bacteria. So even in E. coli, this long circular DNA that has millions of letters, each gene is about, let's say 10 to the 3 letters of DNA, you can say. Human beings much longer. Each one appears once here, with some exceptions. Our cells have 2 copies of each chromosome. So we have, you can say, two pieces of DNA in each of our cell. And there are cell types that have many copies, but typically it's 1, 2, like that. And other questions? It's really fascinating to, as a teacher, to see where I skip, because it's sometimes hard to go back to the, because I was a physicist. Yeah, I studied PhD in physics. I had no idea. The only thing I knew about proteins is what I read in the back of a cereal box. It said protein, you know. Now I know a lot more about proteins, but you have to remember. So it's very helpful, these kind of questions. And biologists, just hang on. Okay, soon we'll get to a point where you...
Is there repetitive processes, or are they destructive? The mRNA is always one mRNA, one Y.
The numbers here. Yeah, so let's say 1 DNA. And now it varies a lot, but it could be, let's say, 10 mRNA molecules. Each one makes, let's say, 1,000 proteins or something. So this could be a typical. So, and these are destroyed very quickly, let's say minutes, and these ones last for a long time. So these numbers, right, it's very important. And okay, more questions? These are fantastic questions. Okay, so we had a glimpse of the biochemistry inside an arrow. And now when we try to think of the entire brain of the cell, we can collect all the arrows there, like all the interactions between transcription factors and genes. And when we do that, we get a graph. So this is the gene regulation network. Gene regulation network is a graph with all regulation interactions on the transcriptional level. There's other levels of regulation. And it looks like this. X1, arrow, X2, right? That's one interaction. But then X1, it could be a transcription factor that regulates X3 and X4. And X4 can also be a transcription factor that regulates X5 and X6. And X6 could be a transcription factor that regulates X5. Why not? And maybe also X2. And X2 can regulate XI, XK, XW, XR. And we get a huge graph like this. And when you try to plot this graph, I say molecular biology started around in the 50s, 60s, looking at E. coli and looking and figuring out this thing I showed you there, this one arrow, how it works, a huge revolution, that there's DNA, RNA, protein, sugar goes in, changes the conformation of transfer, binds the promoter, amazing, activated, this is, and then biologists used this knowledge and found out that the same principle applies in elephants, sea urchins, mice, but different genes and different transcription factors for different signals, right? But the same principle. And around the year 2000, could map an entire organism, also a big chunk of an organism like E. coli, and you plot it out, and it looks like, the technical term is a hairball. Huge graph that you can't understand. It has, and we will understand it in this course, by the way, so, because since 2000, there was a big revolution called systems biology to understand these kind of networks in ways that human beings can understand. So this has, as I said, about 4,500 nodes. Nodes are genes. And around 10,000 arrows. This graph. So this is GRN. In the first part of the course, we'll try to understand this object. What is structure? What it does? How we can think about it? Because in physics and in chemistry, etc., we know that a dynamical system that has thousands of degrees of freedom is usually incomprehensible. Yeah? But in biology, there's something special that happens. It's not just any kind of network. It's a network produced by evolution in order to function, as I said, under very tough conditions. And therefore, as we'll see, it has special features that make it understandable to human beings. Just as an analogy, just like the blueprint of a machine designed by engineers is understandable to human beings, because otherwise they could never build it out of small parts that they can understand. But evolution, it doesn't go to engineering school, but still has some shared principles. Yeah.
You said there were only about 300 transcription factors. Are they the only ones with outgoing arrows?
Transcription factors are the only nodes with outgoing arrows. Between 1 and 10 to the third outgoing arrows. Yeah, we'll talk about that. Each gene has 0 to 6 incoming arrows in this graph. Yeah. This is the GRN, and let's say for E. coli, it's virtually known. To say we can say we have a graph that each arrow, it's not a probabilistic thing or a noisy thing. It's a well-defined experimental fact. Usually, you need to see that X actually binds the promoter physically. That when you delete X from, you can cut DNA, by the way, with molecular scissors, make an E. coli that doesn't have the gene for X. Yeah? That Y expression changes, goes up or down. You can purify the DNA and the transcription factor in a test tube, and so you do a lot of experiments, and you satisfy that there's an arrow there. But that's not, knowing that there is an error there is not enough. We need to know a little bit more in order to do modeling. We need to understand, what I want to say is there's numbers on the arrows to tell you how strong they are. Okay? Not all arrows are the same. So I'm going to erase this, this part here. So we have to say goodbye to it now, this part. And so I have space to draw the more information about this network. And soon we're going towards looking at the equations that describe this network. That's where we're going in this class. And the time scale, how quickly it works, etc. And we'll get to the question of cycles too. Not this lecture, probably. Okay, where are we? Repressor, separation of time scales, gene regulation network. Okay. I think we're entitled to a nice deep sigh of relief. Personally, I think so. So I really thank you for the questions. They help me a lot to understand where you are and where I am. So I want to say something about x arrow y, and that is to say that we have x arrow y. There's what's called the input function, which says what is the rate of y production given a certain concentration of X star, which is the active form of X, the form that actually binds the DNA. Okay? And so on the x-axis, I'm going to draw X star, and on the y-axis, I'm going to draw rate of Y production. I say I'm writing kind of small. Can you see over there? Let's say, ask you there. And this function comes from this binding event here, which is a chemical binding event. And like all chemical binding events, it has a certain equilibrium constant, which is, you can say, the concentration of X star, which 50% binds this site. And so that gives us a typical scale. This is the concentration of X star for 50% activity. And the functions, also I want to say that when there's an infinite amount of protein, X star, it's all going to bind the site. The site is going to be bound for sure, right? So there's, like all biological functions, this one saturates, it saturates. So we already know it has a halfway point and it saturates at a maximum value, which we'll call beta. Beta, the maximal value. And then what can it do? It starts at 0, let's say, reaches 50% here. So what can it do? What it does is this. Okay, this is typically what it does. In this function, when you solve the physics of a simple binding event, basically, you can see that this function here can be described by what's called the Hill function. And this Hill function is that the rate of production, the rate of production of y is beta x star divided by k plus x star. And I'm going to put here power n. Beta is the maximum. When x star equals k, you get half of beta, as you can see here. And this n tells you how steep this curve is. So I drew here a curve, let's say for n equals 4. If n equals 1, it looks like this. And if n goes to infinity, it looks like this. Like a step function, almost. And typically, n equals 1 to 4 in real biological circuits. And this function, this Hill function, comes from, you can say, sometimes you have four sites for X right next to each other, and you need 4 of them to bind in order to get effective production. Something like that. So this is the Hill function. And in this course, sometimes we'll just make the approximation of a step function. If we want to make things really simple, we say if x star is above k, full-blown expression. If it's below k, no expression. If you want an easy approximation. So we need some numbers. Each arrow has, let's say, k and beta and n. We need to know these numbers. Did I explain myself? I see some puzzled looks, but so it's a good time to ask a question if you. Thanks. Is this an empirical fit? So it can be analytically derived from binding equations like partition functions. And it's also a great fit to experimental data. Did I answer your question? So it's great that you have both of those. What else?
Doesn't it die out sometimes?
So does it ever go down? So the question is, when X concentration is very high, can it go down?
Break.
Huh? Break. Usually it's monotonic, and sometimes it's what's called biphasic, go up and down. We'll discuss that, but usually not. Yeah. Interesting, yeah. Interesting questions. Actually, I'm writing down these questions. Who's writing down the questions for me? You, Avi. Because, I'll tell you a secret, in this course, I'm developing the second edition of this book. And so your questions are helping me understand where I'm making the jumps and the skips. And so there'll be new chapters that we'll develop together, actually. If you choose to stay here, yeah? Not making any assumptions. Now, you know, we can now erase this part. What about repressors? Repressors also have input functions, and repressors. So this is for an activator. Why is it for an activator? Because the more activator there is, the more Y is produced. Repressor, the more repressor is, the less Y is produced. So repressors production have this kind of functions, exactly the same. This is their mathematical formula. It's just a decreasing function of XR with a halfway point at K. What happens when two arrows go into a node? When two transcription factors regulate a gene? Okay. Then we need basically a two-dimensional input function. And a lot of times those input functions look like X star, look like a product of Hill functions. Because it's the probability of binding X and Y like that. And they look like AND gates and OR gates. We'll cover that not in this lecture, where you say either both signals are needed or either one is enough. A lot of times that's a good approximation. But I want to say that part, how multiple inputs are integrated, is the unknown part. So I told you the network is known, but these logic gates are the unknown parts. We still don't have a good way in biology to systematically look for them. So there are biologists here who can develop a method to tell me if it's an AND gate or an OR gate systematically. This is very important because we don't know. We only know that for a few cases. And when I started biology was in my postdoc. So I was a physicist and I went to biology in my postdoc, like biological physics. And at that time, it was a whole PhD thesis to discover one arrow. Yeah? And by the time I finished my postdoc, technology developed so much that in one PhD thesis, you can find an entire network with 10,000 arrows because of basically automation of the same experiment and miniaturization on a little chip. So technology advanced hugely, giving us access to seeing the entire brain of a cell, you can say, bacteria, also human cells. And that's one of the things that sparked this field of system biology, because if you have the information, you can think, what new concepts do I need to in order to understand how it works? So, and it's still going on. I mean, now, since I taught it 2014 until now, there was another revolution, where in the past, you did those amazing experiments about 10,000 arrows, but you needed like a few million cells and make juice of them and find the average. But now you can do it from one cell, the little, what is it, picograms of material there. You can do from a single cell. So if your cells are different from each other, like you have a collection of nerve cells and muscle cells and stuff, you can do. So that revolution is now playing out and make amazing. And then you can see all kind of correlations. So biology technology is expanding exponentially, and each new technological advance opens up new questions. So it's far from finished. It's very, we need you. You know, one thing I want to say also is that these biological systems are modular because you can cut and paste in new genes. So for example, and this I want to tell you both because modularity is amazing, and also it's a primary experimental tool used to understand these dynamics. So you can take a piece of DNA from jellyfish, then it codes a protein that's green fluorescent. It makes this jellyfish green fluorescent. It's called green fluorescent protein, like everything in biology, GFP, yeah, green fluorescent protein. So you take this gene, and now you can glue it in the test tube to a promoter from E. coli that responds to sugar. And you take this piece of DNA and make it a circle and put it in E. coli. And now E. coli turns green when you put on the sugar. Isn't that amazing? Did it have to be that way? And what do the E. coli and the jellyfish have to do with each other? Yeah, but that's the way it is. Okay, so it's a modular, plug-and-play kind of architecture, giving the possibility for evolution to over a cell generation time, this thing is fixed. But over hundreds of generations, these arrows can change, rewire, new genes can be added, making this computer on a slow time, so many generations, change especially if it's better for a new condition. Then those cells that have that different network make more babies, take over. So that's even a slower time scale we're talking about. Times of many generations, these arrows are plastic.
All these kind of promoters are modular. I mean, you could take different promoters and always.
So the question is, are all these promoters modular? And to first order approximation, yes. And there's a field called synthetic biology now in the last 15 years that designs circuits inside cells built based on parts. So you can make cells oscillate, counters, multipliers, and stuff. You can build circuits into cells. And you can read out with green fluorescent protein, which is what I did when I started my lab, read out for all the genes in E. coli, made bacteria that turn green fluorescent. And on a little robot, you can now have thousands of bacteria. They're all identical, but each one turns green fluorescent according to a different node here. And you can get a readout of the entire brain of E. coli optically, adding a little copy of the promoter with green fluorescent protein, not touching the rest of the DNA. So that was exciting tool, and that's how we know a lot about the dynamics, which is what I want to tell you now. Any more questions? So we're doing on time. Yeah. You know, my goal is to finish at 12:30, and no break to finish at 12:30. Okay, so if you really need to go to the bathroom, you can do that during the talk, but you get half an hour free for your life after this. We're coming to the grand finale of the first lecture. Talk about the equations, right, that guide this network. So we talk about equations for the dynamics of a single arrow and next time we'll talk about circuits made of many arrows, several arrows. So I want to just talk about the basic equation of one arrow. What's the time scale of signal comes in and protein Y is made, this kind of interaction. So this equation looks like this. So we have here, this is the rate of change. Oh, I wrote x arrow y, right? What are x arrow y? So let's write down here y. Sorry. Rate of change of y concentration. And it's a very simple equation. Beta is production rate. Now, what I'm going to do I'm going to assume situations where either I put a lot of the signal, so a lot of activity, or zero. So it will be either 0 or it's beta here. So if we're in a situation with a lot of signal, a lot of input signal, we have production rate beta, protein, concentration per unit time. And if that was the case, Y would just be made, and the cell would explode. We don't want that. So Y is also removed. And this removal term is almost always a kind of like radioactive decay. You have a certain probability per unit time for this protein to be removed. Now, what do I mean by removed? This removal rate is made of active degradation which means that there's a machine, a proteasome, that takes protein Y and cuts it up back into amino acids the cell can use again. That's the rate of active degradation. Okay? And dilution by cell growth. What does that mean? If I have a cell and there's some proteins in it, and the cell now grows and doubles, but I'm not making any new proteins, then concentration is going to go down per volume. So that's basically linked to cell growth. Dilution by cell growth. Any questions about that?
Why would they both be linear and why?
What?
Why would they be linear?
Why is this removal rate linear and why? So from the point of view of dilution, this is just basically the rate of volume change divided by volume. This is just how much volume is expanding. And degradation is basically these Pac-Man proteasomes find the protein and degrade it. So it's like a certain rate per protein unit. So that's like a chemical reaction, or a first order reaction where you have each protein has a lifetime because of this degradation machinery. That answer your question? So this alpha has units of basically 1 over time. Y over time, because dy dt. And this has protein, let's call it concentration over time. These are the units here.
Yeah, but wouldn't it depend on the rate of concentration between alpha and y? Because you have to keep it in a certain relation for it to be like a percent of y.
You have to keep it in a relation. Can you explain? Say more?
Yeah, let's say that you have like 1 alpha for every 10 y.
Oh, so yeah.
If you have to keep the ratio between alpha and
Got
y
it.
to be in a
Okay.
certain
Yeah. So you're saying you have to keep a ratio between alpha and y. And I want to say, just maybe explain myself. Alpha, it's not the concentration of a proteasome or something like that. It's the concentration of it's assuming here that the proteasome machinery is unsaturated. There's many, like Y, it's taking up only a small part of its life. And then this gives a basic half-life for Y to be destroyed. You can make a more complicated model where now, if there's a lot of Y, it saturates that machinery. But we don't need to right now.
So it seems to me that it assumes that there is a machinery that keeps alpha, or the machinery behind alpha, at a certain level with relation to y.
Yeah, so there's a machinery that degrades Y, and it has a certain rate, and we'll call that rate alpha. That can change between conditions too, because this machinery is made of proteins. Proteins are expressed by other proteins. If this cell is stressed and a lot of proteins are unfolding, it makes more of this degradation machinery, for example, to get rid of those proteins. So there's definitely, these beta and alpha numbers depend on the situation. Thank you.
Why is it density? Why is it?
Yeah, it's concentration. Y units of Y are concentration. So we have the units, concentration 1 over time, and concentration over time, concentration over time. We're fine with the units? Good. Okay, great. So and maybe there's more questions. Soon we're going to do I think we'll do the reverse little discussion. So get ready for that, where people who are math people can talk to people who are more biology people. But let's just get a sense for the solution of this equation.
There's no independence
On x, yeah. So great, thank you. So is there a dependent on x? So where would dependent on x be? X is the signal that activates it. So in principle, it'll be here. This function here, let's call it now f of x star. And I now made a jump because I was too quick to say that I'm going to assume that when x star is present, it's all the way over here, so we're at the maximum beta. But in principle, here should be f of x star. Like this input function depends on the signal. Am I clear? I'm worried. No, there's no steady state here because y can change, function of time. But let's start with steady state, if you already asked. Okay. So suppose we have a lot of signal, and we have a maximal production of this rate of this protein at rate beta, and we have removal at rate alpha y. What is the steady state? So when we let the cells grow in this condition for a long time, let's say 10 generations. So what is the steady state? Steady state level of y happens when dy dt equals 0, And this means that 0 equals dy dt equals beta minus alpha y. And the solution is that y steady state is beta over alpha. Now let's check the units. Concentration divided by time divided by 1 over time. So this is concentration. Right? What does this mean? Very easy to interpret. If there's more production, beta is bigger, we get higher steady state value. If there's more removal, we get less. It's the ratio between production and removal. So that's a steady state level. Am I clear? Now let's look about the dynamics. So what I'm getting at here, so we talked about degradation, dilution, steady state. What I want to know here is how long it takes it to reach a steady state. Once I put on the signal on the cell, how long does it take it to reach a steady state level? And the steady state level, of course, is very important. It's the typical concentration at which this y needs to work. So evolution makes sure to tune this beta and alpha so that this concentration is the right one. So if it's a transcription factor, maybe it's 100 copies per cell. If it's a major structural protein, like a skeleton, maybe it's 100,000 copies per cell, etc. So beta and alpha are tuned for each protein. How are they tuned? By mutation, the letter changes in those binding sites I talked about, of RNA polymerase and X. Make a little change there, and RNA polymerase binds more or less, changing beta and alpha. So see, so that's the way evolution can tune those rates. And the cell with the best beta over alpha makes more babies. But how quickly, right? So how quickly can you reach this functional concentration? How long does it take? Yeah. So how long? So what we want to know is the response time to reach y steady state, the functional concentration of y. So how long does it take? And in order to do that, we need to solve this equation. So I'm going to start first from the case of we start at y steady state, and suddenly beta goes to 0. So you take away the sugar. That means the steady state is 0. But how long does it take to get there? So dy dt equals minus alpha y, because there's no beta. And here the solution is, this is t, this is y. We started y steady state, and the solution of this equation is y steady state e to the minus alpha t. And you get this. Right. In order to understand what the response time is, I'm going to be like an engineer and say what's a typical time scale for this reduction. Let's ask how long it takes to reach 1/2 of y steady state. Call that t1/2. So t1/2 time to reach halfway of the change, whatever change we make, let's say from state to 0, what's the time to reach halfway of the change? And if we solve for that, we see that we want y of t1/2, we want that to be 1/2 y steady state, which is equal to y steady state e to the minus alpha t1/2. So I'm plugging in t1/2 there. And y steady state falls out, that's nice. And we get that t1/2. So I need to take a log here. So we get that minus log 2 equals minus this alpha t1/2. And I get that t1/2 equals log 2 over alpha. So that, so it doesn't depend on production rate. It depends only on the removal rate. So, steady state depends on both, but response time depends only on removal rate. It's the eigenvalue of that equation. And let's do the other case is when beta goes to 0 and suddenly we add a lot of the signal. Beta goes to some value, beta. The solution in this case looks like this. It reaches y steady state, and y equals y steady state 1 minus e to the minus alpha t. How long does it take it to reach halfway? Response time, t1 half equals y steady state 1 minus e to the minus alpha t1 half, which equals 1 half y steady state. Yeah, right. And the answer is the same. So no matter what you do, add, remove, no matter how strong the signal is, the response time, the time you need to get to the functional level, is determined by the removal rate, not by the production rate. How high you get to determined by production and removal, but how fast you get there, determined only by the removal rate. So how much is this log 2 over alpha? And I want to show you that for most proteins in E. coli and also in human cells, it's really very slow. It's too slow. I want to explain why to you. I want to say most proteins are stable and have virtually no degradation rate on the time scale of cell generation. Maybe I should mention that a strategy to get fast response times, that's with small response times, is to make alpha very big. Right? If I make removal very fast, for example, if I make this protein very unstable and destroy it, alpha will be very large removal rate, and I'll get very fast responses. That's somehow a little bit maybe not intuitive, but you can get fast responses because in that case, in order to make beta over alpha, your y steady state, you need to make you need to produce a lot and to break a lot. So you break. And why is there fast responses? Now suppose I stop making it, then because I get it? So you can have fast response times if you make your protein unstable and degrade it very quickly. But most proteins aren't like that. Most proteins in E. coli, in human cells, etc., last an entire cell generation. In human beings, it's maybe half of the proteins. In E. coli, it's 90% of the proteins. They last much longer than a cell generation. And you can also think, why isn't it such a great strategy to make, break? Even though maybe nowadays, people who make machines want them to break very quickly, so we buy new ones, right? But cells don't do that. Maybe we can learn from them. Because making proteins costs resources. Yeah? And if you spend your time making a lot of proteins, it costs ATP, costs energy, costs amino acids, then a cell next by that figured out a better way to do this is going to grow faster. So there's a cost. And that's why most proteins are stable. You make them, you don't break them. And then alpha is only the dilution. And I want to tell you that this dilution is very long. It's log 2 over a cell generation time. And why is that? Imagine that you stop making a protein, and you have a cell that has a certain amount of protein, y steady state. And this cell grows twice the volume and then divides. Right? So this is volume V, this is 2V. This is one generation. The cell is doubled. Each half will have y steady state divided by 2 because we're not making any more proteins. Cell divides. So after one generation, concentration has gone down by a factor of 2. So that's exactly T1/2. So that's the dilution rate. The dilution rate is log 2 over the cell generation time. If you have a stable protein and you stop making it, it'll grow down to half the concentration after one cell generation. And I told you the response time of making it is the same as breaking it. So also if you have none of this protein, now sugar comes in, you need to make that pump, it'll take it one generation to reach halfway to the steady state level. One generation. That means you're working for your children or your grandchildren. Only they will get the Y steady state. Get it? So these are somehow, I hope you get the feeling, weirdly too slow. So yeah, so T1/2 for stable proteins is one cell generation, which is very slow if you want to enjoy life with your children and not only your children. I don't know how many of you are parents. So we have a dilemma, kind of, how can this be, how can cells function, because cells function faster than cell generation, how can they resolve this problem? And that's going to be in the next lecture, when we'll look at the circuits in this huge network that I've erased, find a really commonly occurring circuit, think what it does, and see how evolution found a way to overcome this problem, making fast response time without the cost of a futile break and make cycle. That'll be the topic of our next lecture. Let's take a nice deep sigh of relief. Thank you very much for all the questions. See you next week if you so choose. Goodbye.